Chapter 2, page 37 in this 7th edition of Kubi Immunology. We're talking about cells of the lymphoid lineage and how they regulate adaptive immune responses. There's lots of things in immunology that seem like a different language that can intimidate students a little bit, but don't let it intimidate you. You need to kind of embrace some of the terminology and learn the language, and then you can learn anything you want to about immunology once you have the basics down. Lymphocytes are white blood cells, and they are the principal players in adaptive immune response. There's some non-adaptive immunity, things like your skin, tears, earwax, things like that are going to block any kind of bacteria from getting in, any kind of pathogens from getting in. But then there's some white blood cells that can learn to recognize specific pathogens. You have lots of different aspects of the immune system, and some of them are adaptive and some of them are more general protections. So now let's look at certain things like here's cluster of differentiation. That's a term that's put on a surface protein that's found on specific cells. So each cell can be separated into different clusters. For example, two of the most common ones that we'll talk about are going to be T helper cells and cytotoxic T cells. T helper cells, as you probably have already read, help to regulate the immune system. They give permission for virgin or naive B cells to go through clonal expansion. They have a T cell receptor, but they also have a CD4 protein on the outside. That's a glycoprotein that's unique to the T helper cells. So what happens is the T helper cell receptor will bind to an M8C carrying an antigen. And it requires kind of this double signal. It's almost like having a handshake with your right hand, but you use the left hand to reach up and pat somebody on the shoulder. So you're reaching around to pat them on the shoulder, and that gives two different signals, the handshake plus the pat on the shoulder. Well, the CD4 is the protein that's reaching up to the shoulder and allowing a secondary signal to go through. Well, if both signals go through, then it signals some effector function in the T helper cell, which will release chemical messages for the cell that it's in contact with. Now, T helper cells tend to bind to M8C class 2 molecules, where cytotoxic T cells tend to bind to T helper cell type 1 molecules. The T cell receptors very similar in both cases, but the cytotoxic T cells have a CD8 protein that reaches up and pats the shoulder. Then you have B cells that have receptors, and they're called receptors until they're released as antibodies. I don't make up the terminology, I just have to teach it to you. Once the B cell becomes activated, gets permission from a T helper cell, it goes through a transformational process, and you can get some plasma cells. Plasma cells are larger than the virgin B cells. They have a lot more rough endoplasmic reticulum because they're making lots of antibody. And they're usually very short-lived. A couple of weeks is about the maximum age of most plasma cells. Now, some of the B cells, as they go through that clonal expansion that you've been reading about, will become memory cells, and they can last for 20 years or more floating around through the bloodstream. So it's really fascinating to look at the differences in the cells and how they very quickly differentiate. Now, natural killer cells work a little differently than cytotoxic T cells, but they do the same thing. They cause normal cells to go through apoptosis. A cytotoxic T cell is going to look for M8C plus antigen. So it's going to be reading the M8C class 1 molecule on the outside, and it's looking for self-antigen. As long as you've got the right name tag, the right M8C, plus the right antigen, the cytotoxic T cell won't kill you. But if it happens to be holding a viral protein because the cell's infected with a virus, then you're going to get the cytotoxic T cell stimulating apoptosis. one of my favorite words, learn apoptosis. It's programmed cell death. It's going to kill the cell to prevent the virus from being able to replicate and spread. Natural killer cells count the number of MHCs on the surface. So if the virus has downregulated the production of MHCs, then the natural killer cells can come along and take them out. So natural killer cells do a lot to prevent cancer cell formation and to prevent intracellular parasites like viruses from being able to spread through the body. Learn the difference between the two. Look at some of the characteristics. Kind of learn what a clone is. If you have a B cell that has one type of receptor, one type of antibody on its surface, it's called a receptor at this point, and it gets stimulated to go through clonal expansion, then it's going to make hundreds or thousands of copies of itself that carry that one type of receptor. So each one of those is called a clone. Some of them can be cells that go out and do something, so like effector cells, they carry out very specific functions, and some become memory cells. And the memory cells are there in case you come into contact with the same antigen again in the future. Again, don't get so tied up in knots over the CD4, CD8, CD28, CD whatever. Those just mean that it's some receptor protein on the surface of these blood cells, and they're used to differentiate them. If you are learning about a specific organism, like if you're looking for Epstein-Barr virus, CD21, or CR2 is another way to call it, these are receptors for complement and Epstein-Barr virus. So if you're working with that, you'll learn that. I don't expect you to learn all of them, but you do need to know the difference between the CD4 and the CD8s, because later on we're going to go into a lot more detail. All right, I will stop at that point, and next will be T lymphocytes.